Hey everyone! Okay, so my second video for this week is going to touch on enzymes. And so I wanted to just go over, again, what are enzymes and why are they important in biological systems. So remember that enzymes are proteins, right? And we have seen proteins before, and we'll see more of them later. And basically, proteins are one of the major class or group of biological molecules or macromolecules, right? These big molecules. And they're made of amino acids, right? And amino acids are just kind of the building blocks. They're these smaller molecules um, that of that are made up of carbon, oxygen, nitrogen, and hydrogen. I'll just kind of quickly sketch out just the basic building block part of it. So you have oxygen, hydrogen. This is a double bond here. Hydrogen here. Nitrogen and then some more hydrogens. And so this is the kind of the general structure that all amino acids, right? We have 20 amino acids, and they all have um, this kind of basic structure here. This is the same across all 20 amino acids, and but then what makes them unique is this what we call an R group here. So there's 20 different kind of groups that come off this kind of basic structure here that make these 20 amino acids different, right? So just to give you a little peek at what those look like, I just grabbed a website. So again, this blue part is kind of the, I'll zoom in on it. Okay, so this blue part is, again, the kind of building block part of it. It's the same across all these different amino acids, but it's this yellow part is what we call the R group. So you can see the R groups vary quite a bit, and it's really these different groups that make these amino acids different. Okay, so you can see, some of them also have some sulfur and some more nitrogen and things like that. So it's really these kind of groups that dangle off kind of the main part that make these amino acids different. And so basically what happens is these proteins, when you string a bunch of these amino acids together, they form a structure. So if you kind of look at, if you imagine you make a little chain of amino acids, so each of these little dots are amino acids. You string a bunch of amino acids together, and depending on what order they're in, which amino acids you use, which R groups they are, they start folding. And so they start kind of folding in these either pleated sheets, or sometimes they make a little helix. And then they have a nut. If you kind of zoom out again, they have these kind of, then they fold even more. They have these kind of bigger, what we call tertiary structures. It's just kind of the different levels of structure of proteins. So basically, it's really about amino acids in chains, and the number and the types of amino acids and the order they're in will create these complex foldings. Okay, and so then they kind of fold in these complex ways, and then you can actually have, it turns out, you can have a couple chains of amino acids grouped together to kind of make your final protein. Okay, so that's just a general overview again of proteins. Okay, and so this folding turns out to be really important. Okay, so let's see. So that was, again, just about proteins. And remember, enzymes are a group of proteins, right? They're a type of protein. So enzymes, what are enzymes again? Why? So they're types of proteins, but what do they do? What's their function? Enzymes. So remember that folding, why I brought up that folding is because the shape and the shape that they fold into is really what's going to affect their function. And what is their function? Well, their function is to catalyze reactions. Catalyze chemical reactions. And catalyze chemical reactions. Okay. So catalyze just means to speed up or to kind of facilitate a chemical reaction. So molecules that are dissolved in aqueous solutions, or basically water, basically they kind of just zoom around and float around, and if two of them bump into each other with enough force and at the right orientation, they can react together. You can kind of think about this like a box of Legos. If you're to take like a box of Legos and shake it, eventually two might hit each other at the right angle and with enough force to fit together. But it would take a lot of shaking and a lot of time, right? So, and actually, just a side note, this is why increasing temperature tends to increase chemical reaction rates, because it speeds up those molecules. So they have more energy when they're hitting each other, more force, and they also hit each other more often. Okay, but this is, you can imagine that this is not the most efficient way of, going about chemical reactions, especially if you're in a biological system where you are kind of reliant on these chemical reactions to take place. So what we do is we have these catalysts called protein, or these enzymes that our cells produce, and that can actually facilitate these chemical reactions. And so what happens, and I'm not going to draw this because I found things that are have better illustrations online. Okay, so remember, so their folding is really important because what happens is their shape is really important because it affects their structure. So what happens is, so here's like an example of an enzyme, right? It has a certain shape, and these might be two molecules that would have to kind of meet and react with each other. And you can imagine if they're just kind of bumping around the place, they may not. It might take a while for them to just randomly bump into each other the right way and with enough force for that reaction to occur. But what enzymes do is they kind of grab those molecules, what we call substrates. You don't really need to know this, but they grab them and they orient them in the way that they need to be oriented in order to actually react together. So they kind of, instead of shaking the box of Legos, you actually grab the two that you want and you put them together in the way they fit together, right? You orient them in the right way, and so then they can react together, and you can get some product that you want. And it goes the opposite way, right? You could start out with something that you need to break down, and the enzymes actually will then help break down those into smaller parts. Okay, so that's kind of like how enzymes help facilitate reactions. And we say facilitate because, remember, enzymes are not actually used or changed in the reaction. They just kind of help it along. Okay, so enzymes are really important in speeding up chemical reactions. Okay, so I wanted to bring up just really quickly, there was something really cool. A couple weeks ago, I saw an article online. This is Scientific American, and basically it was this really neat article I saw a couple places about how online gamers actually have been our scientists and engineers have kind of worked together with online gamers to help solve protein structures. So it turns out that for decades, basically, there's this HIV, there's this protein that scientists and engineers had been using computers to try and understand the structure of this protein, which would be useful in fighting AIDS and HIV. But computers are not quite as good at spatial reasoning than humans. So what they did was they created this game called Fold It. It's actually an online game, and they recruited gamers, and it's you play this game, you get points and things like that, and you basically simulate protein folding. And it turned out that within, I think it was 3 weeks, so they've been working on this problem. This protein cutting enzyme, produced by retrovirus, similar to HIV, and they've been working on it for about a decade, and working with these gamers, it took the gamers about 3 weeks to solve this protein structure. And so that was just a really cool, neat, application of understanding how enzymes are important and how protein folding is important. And I'll just go to the website called Fold It. So if anyone has any interests, if you're a gamer and want to, help save the world and cure disease and things like that, here's the website, Fold It, and you can actually join and help understand and solve these puzzles. It's really cool. And also, I just wanted to show you, it's also just a good resource just to review. If you just click here, basically, it takes you, the science behind Fold It. So it talks about what is protein folding, So again, it's important about the structure. It talks about what amino acids are, what shape proteins fold into, why is shape important, and then it talks about some of these different proteins. So I just thought it's a good resource in case you want to review, and just kind of cool. So yeah, so you can go and become go help cure cancer and stuff like that. So that was cool. Anyway, so that was just a neat application I wanted to bring up. So yeah, just thought it was interesting. But anyways, moving on. So yeah, so enzymes again, they're here to we produce enzymes to catalyze these chemical reactions, right? Okay. So I mentioned earlier, and Dr. Pearson brought this up, is that so temperature tends to increase these chemical reactions rates, right? And so if you actually plot temperature along a graph and reaction rates, right? So as you increase temperatures, the reaction rates of these chemical reactions tend to increase, especially even with, particularly with enzymes too, because the faster these molecules are moving around, the more often they're going to bump into the enzymes, and the enzymes can grab them. So if we plotted temperature with reaction rates, you would see an increase, right? You would see, okay, as temperatures increase, you also have these reaction rates increasing. And so you might ask, okay, so why don't we just produce a lot of body heat and have really fast chemical reactions? There's another side of this graph. So if we're using enzymes, something happens at a certain temperature, and then you actually start getting a decrease in the reaction rate. So what's going on? What's different? What's important about this temperature? Okay, right here, right? What's going on here? Okay, so let me ask you, have you guys ever cooked an egg? So if you've ever, hopefully you have cooked an egg at some point in your life. If not, it's about time to get started, right? You're going to have to start feeding yourself at some point. So if you first crack an egg, you have the yolk, which is yellow, right? And then you have the white of the egg, which I don't know what that's called. Sorry, someone can look it up and leave a comment if it's that important. So you crack your egg, right, on the frying pan, and when it's cold or before it cooks, it's clear, right? It's you can see through it basically. But as you cook, you'll notice that it turns white, right? So what's going on? Why does your egg go from clear to opaque, or from clear to white? Well, it turns out that the white of the egg is where all the protein is. Okay, it's just a lot of proteins, and so what happens is as you apply heat to proteins, remember those really intricate folds. If you apply heat to those folds kind of fall apart, and that's what we call denaturation. Let me write that somewhere. Denaturation. Okay. So denaturation is caused by high temperatures. Also, it's caused by extreme pHs, right? Acidity and basicness, or alkalinity, right? High pH or low pH, depending on the protein, can also cause this denaturation. And basically, what it does, again, you remember structure is really important for function and for the protein properties. So if it loses that structure, its properties are going to change, and its function is going to lose its function, right? So again, that's why when you cook this egg, all these proteins are being denatured. They're losing their structure, which originally allowed light to pass through, and now after it heats up, it loses its structure and then no longer allows light to pass through, and that's why it turns white like this. Okay. The same thing happens if you cook meat, right? You go from kind of reddish to these kind of brownish color, right? And it looks a little bit different. The texture is different, right? And that's, again, you're breaking down these kind of structures of proteins. Okay. Okay. So I'm going to leave you with today's application question. And today's application question is, when an invading bacteria or virus infects your body, one immune response is to raise your body temperature. This is also known as running a fever. So how does this protect us from infection? Why is this an adaptation we have for fighting infections? Also, why are you in danger if your fever runs too high, over 105 degrees? Why does that put you in danger? Okay, so think about what you've learned today. Think about what you know about enzymes and proteins, and generate some hypotheses about why we have this immune response of running a fever, and then why it's dangerous that if it gets too high. Okay, so leave comments. If you have questions, also feel free to leave comments. See if someone else can help answer them. And if there's something that wasn't totally clear, please feel free to leave comments. Okay, all right. Thanks, guys.